All right, so next we're going to talk about the process of disease, okay? And we're going to chat about the main stages of disease. So disease process, any disease process, starts with the very first step, which is adhesion. So regarding adhesion, I want you to know the main adhesion mechanisms for bacteria and for viruses, okay? So in bacteria, we're talking about fimbriae, adhesion pili, okay? Special proteins called adhesins, capsule, slime layer. Now, I'm gonna harken back to the discussions that we had in our very first unit, very first. Remember, we were talking about phenotype and how gene expression defines phenotype. Let me ask you a few questions. Presence or absence of surface proteins adhesins. Adhesins, are they encoded in DNA? Yeah, they are. Ability to make a capsule, is it encoded in DNA? Yes. So if bacteria have genes for certain virulence factors, all of these are virulence factors. Does that make sense? Okay, so all of these are virulence factors. Now let me ask you this question. I want you to start thinking in the context of what makes this microorganism so virulent or more virulent than that one. Are you following me here? So let's say you have two strains of Streptococcus pneumonia. One strain has a gene cluster that allows it to produce capsule. Which one will be more adhesive? One with a capsule, right? The better it adheres to cells, the better it is capable of establishing an infection. So the strain with a capsule will be more virulent. Does that make sense to you? You got it? Now remember we were talking about the gene transfer between bacteria. Remember? Can those genes potentially be transferred from one microorganism to another? So virulence can be transferred, can be transmitted from one microorganism to another. They can acquire virulence factors. They can randomly become, that particular cell can become more virulent cell. Are we clear? Good. And for viruses, the mechanism of adhesion is receptors. And this is something absolutely fundamental. No receptors, no infection. Interestingly enough, it's not the other way around. So, cells that do not have a receptor will never be infected. This is basically a determination of the virus, tropism. The word tropism means where the infection is. Okay? Are you following me? So, but take coronavirus. Its receptor is the protein called ACE2. It is found on the cells in the lower and upper respiratory tract. This is where virus infects. Does that make sense? It's also found on endothelial cells in the blood vessels. There's no evidence towards infection of endothelial cells, just none. So, if there is no receptor, there's no infection. If there is a receptor, there may be infection. Am I clear? Good? Penetration. There are a gazillion of mechanisms how viruses, fungi, bacteria can penetrate. So what I want you to understand, infection can be intracellular or in the tissue, extracellular. Does that make sense? Remember how we said that viruses are obligate intracellular parasites? Well, they are. So we can say under intracellular always for viruses. Now for bacteria, it can be either way. Okay? They can stay outside of the cells, they can get into the cells. For different bacteria it's different. Am I clear? I just want to highlight that pathogen, after the adhesion, has to penetrate, has to get either into cell or tissue. Are we clear? Good? Which brings us to a really, really long conversation about the virulence factors. And we're going to start with, so basically what we're going to talk about now, we're going to talk about actual pathogenesis. Okay? Sorry, misspelled. And the first type of virulence factors are going to be exoenzymes. We mentioned those before. Those are enzymes produced and released out of the cells. They may end up in the tissue. They may end up in the cells. So there are several types. DNAs, glycohydrolase, proteases. Yeah, there are 4 types that I want you to know. So, glycohydrolases, proteases, nucleases, and phospholipases. Okay, now, do you need to know the examples? No. What I want you to understand, what they target. So, glycohydrolases target carbohydrates which comprise extracellular matrix. Does that make sense? Now, let's pause for a second. I think I'm going a bit too fast. Now listen. Cells in your tissues are held together by extracellular matrix. Clear? If you have an enzyme that destroys extracellular matrix, the mortar that holds your cells together is gone. Cells start to fall apart. Okay? Proteases. Well, duh. Proteins. For example, collagen in the connective tissue. Collagen is the main component of connective tissue. If it's gone, connective tissue falls apart. Nucleases. Usually DNA, sometimes RNA. Let me pause here for a second and highlight what happens here. So we're going to talk about it. I'm going to mention it briefly when we will move into the immune system, but look at this. When your cells called neutrophils fight an infection, are you listening? Are you following? Fight an infection. they die. When neutrophils die, the DNA gets released outside, and DNA strands wrap around bacteria. This is called neutrophil extracellular trap. It's going to be in the immune system. It's going to be later. Does that make sense? So basically, cell becomes trapped like in the net. Cells like Staph aureus can produce nuclease, DNAs, that will destroy those traps, and the cell can swim away, kind of. Got it? Finally, phospholipases, obviously they target phospholipids. And therefore membranes. Good? So, this is just one type of virulence factor. Second, so we're going to talk about three big concepts in virulence factors, which are exoenzymes. Now we're going to talk about toxins, and after that we're going to briefly mention mechanisms for immune evasion. So let's take a look at another aspect of pathogenesis, toxins. So toxins that are produced, and again remember, toxins are produced because there's a gene for making toxins. Toxins can be divided into exotoxins and endotoxins. So let's take a look at the difference. I'm going to start with something that is already familiar to you from our previous conversation about gram-negative microorganisms. Endotoxin is a lipopolysaccharide. Remember that molecule that can be found in the outer membrane of bacteria, LPS? Now, if you remember, LPS is present only in gram-negative bacteria. Okay? Now, what are the features of LPS? Well, let's start with, what about the lethal dose? LD50 for LPS is very high. You really need a lot to kill a person. Okay? It's very thermostable. So even boiling cannot get rid of LPS. Kind of running ahead of a train, I can tell you that the only way to get rid of LPS is to filter. You can filter it out. Okay? There is no specific... Okay, no, that's a bad statement. No antibodies produced. Now this is a really important thing. So I'm gonna go on a digression, a little digression here. There's a guy in Australia who breeds taipans. Do you know who taipans are? Taipans are most venomous snakes in the world. Like, we're talking about like 1 minute from bite to death. Okay. So this guy very early started to inject himself with taipan venom from very, very small doses, slowly escalating the dose. Since the venom is a protein, the toxin is a protein, his immune system developed a very robust antibody response. And now he can basically just, taipan will bite him and nothing happens because he is immune essentially. Make sense? And the toxins aren't like that. You get exposed to lipopolysaccharide, for instance, as the result of gram-negative infection, your immune system doesn't form an adaptive response, it doesn't form memory, doesn't form antibodies. 2 weeks later, you can get exposed to a different gram-negative microorganism, like previous exposure never happened. Does that make sense? Now, the symptoms of endotoxin exposure, I can say is a general systemic inflammation. So basically you feel like crap. You have fever, headache, your joints, your muscles, your bones, they all ache, all that kind of stuff. But there's no specificity. Whether you're exposed to LPS from Salmonella or Shigella or E. coli or Neisseria, doesn't matter. Am I clear? Now let's compare endotoxins to exotoxins. So structurally they are usually proteins, can be produced by both gram-positive and gram-negative microorganisms. They usually have very low LD50. You may remember we discussed this for Clostridium botulinum. They can be thermolabile. Not all the time, but most of them are thermolabile. And they cause a robust antibody response. And finally, they have very, very specific, cause very, very specific symptoms. Okay? So, in the lecture notes, I inserted this table there. In the lecture notes, there's a table for with different types of bacterial toxins. Okay. Do you need to know? You don't need to know an example. I'm not going to ask you how does botulinum toxin work, okay, or how does pertussis, Jesus Christ, titanospasmin works. That makes sense? Like, I'm not going to ask you this. You need to know 3 types of exotoxins. Intracellular targeting, membrane targeting toxins, and superantigens. What kind of question you can expect here? Look, intracellular targeting. I give you an example. Clostridium botulinum blocks the release of acetylcholine from motor neurons. Targets very specific cellular mechanism, intracellular targeting. Does that make sense? Toxin of Clostridium perfringens destroys the membrane of connective tissue cells. Membrane targeting toxin. Toxic shock syndrome, SST of Staph aureus induces abnormally high level of immune response, superantigen. Match the description with the term. Am I clear? Okay, those questions usually do not cause any troubles. So far, so good? Okay. Terms that you need to know, I'm not going to put them on the board. Toxemia, toxigenic, toxinosis, intoxication. So, toxemia, toxin in the blood. Toxigenic, microorganism producing toxin. Toxinosis, disease caused by a toxin. Intoxication, ingestion of a toxin, just terminological. Okay? Now, last bit about pathogenesis. We're going to talk about immune evasion, and then we're going to take a little break. This can be a semester-long course on immune evasion. So what I want you to appreciate, what can microorganisms do? You need to, what is the role for capsule? Macrophage evasion. What is the role for nucleases? Breaking down antibodies. What is the role for leukocidins? Killing white blood cells. Well, complement inhibition is self-explanatory. Interferon inhibition is also self-explanatory. Okay, they inhibit complement and they inhibit interferon. And last two mechanisms, intracellular infection and one of my favorite, molecular mimicry. I'm going to stop for a second on this concept right here. So what is molecular mimicry? In this case, imagine you have an organism, schistosoma, say schistosoma mannsoni. Okay, you follow me? Tiny little fluke. Okay, look. And it covers itself with human proteins. So immune system doesn't see it. It's like the wolf in the sheepskin. You see what I'm saying? But that's phenomenal. Now, what do you need to know here? Oh, come on. Description type, or what do leukocidins do? What does capsule do? What is the role for nucleases? Just very, very simple, very basic stuff. Okay?